a doctor and a biology and a biochemist and a cytologist, epidemiologist, uh, anatomy physiologist, uh, studies basically and a pathologist and a medical therapist or a biomedical medicine person, someone who works in the development of medication or medical therapy. They all study mostly like polymers, anything to do with physiology, uh, cell structures, and the functions of the physiological functions and the pathological dysfunctions of those cell structures and polymers, and even their quantum physical energy statuses, like whether or not they're ionic or how they have partial energy, how they transmit or have transmission, uh, how the system works entirely, which is basically a normal functioning system is all about physiology. Well, guess what? It is inconclusive. It is inconclusive. Physiology is today. Our government is saying that the proteome of a human cell or the human body could possibly be in the lines of 10,000 up to billions of different proteins, and they're not sure. And that's the latest from NIH Gov Web. And, how to trace these out to DNA, basically, is another thing that has to go on. All of these things are not known currently today. And now I also want to talk a little bit about the difference between polymerases in the genetic field or the medical field versus using viruses to edit DNA, like CRISPR versus a virus. CRISPR versus a virus. That's major difference there. Polymers and polymerases are major constituents to cell structures of any kind. And, you also have your molecules and ions and very light polymers. But still, the difference between these polymerase genetic modifiers or any kind of cellular polymer modifier versus using viruses is a different structure. Basically, your polymerases that are already in your cells that perform biogenesis and mitosis, they're already there. A virus basically inserts DNA so that those polymerases can insert it into your new mitotic cell. And then when it does that, you have new DNA, and it can cause either a good thing or a bad thing or nothing. Any of the three. So, CRISPR is a little bit different. It's a polymerase that actually can actually build DNA. If you really want to modify CRISPR, it can build either DNA strands or genes or particles, or it could build it could possibly be modified someday in the future to build all sorts of like entirely a whole genome or a whole DNA structure, maybe an entire chromosome, because our DNA is essentially made from polymerases. We absorb nucleic acids. They travel into the cells eventually, somehow. If they're absorbed, they go into the nucleus, and our polymerases build them. And we have DNA structure that then produces an RNA structure and certain proteins that cause ionic reactions to occur specifically based on different patterns and polymers and ions, and it occurs more precisely, but however, there is a law behind it. There is a law of diverse reactions or diverse chemical bonding. Okay, and this can be ionic or it can be negative. It could be partial, could be temporary. There are such things as temporary reactions. They affect human physiology and pathology. So that's one thing. So this is pretty much the video on the gist polymerization and cell functioning.